Bernice Mosers. She started here in fall of 2003, finishing her undergraduate degree. She started working with me with the Xenophus laevis frog as part of her undergraduate degree. And she became interested in toxicology and interested in how caffeine and pseudoephedrine interacted on developmental toxicity. During that time as an undergraduate, she received 2 grants, one from Beta 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 and one from Alabama Academy of Sciences. She finished that project as an undergraduate and has her results published in Bios, so did very well in that. After she finished her undergraduate, she began working with me on her master's thesis, and she had attended several scientific meetings with me, to toxicological meetings and other areas, and she became intrigued with potential compounds in the environment that had estrogenic effects, and particularly in amphibian populations, very popular at the CTAC meetings that I attend. This project has had a lot of challenges. The work she has presented today has accumulated a tremendous effort on her part to get all of this done, and I'm pleased to introduce to you Bernice Mosers and her talk on estrogenic effects in amphibians from local watersheds. And, I'm sorry, I just said that and messed up. At the end of the talk today, after we are done, we have a general seminar. We'll do our general question and answer session. Then Dr. Hamasu, myself, and Dr. Klein will take her up to do her defense, which I'm sure that she will do well in. And we will have, hopefully, what will be an award celebration at 5 o'clock today at Jefferson's. So you're all welcome to attend that if you would like to come for that. And I'll remind you at the end for that too.
You paying right now? Yeah, you're paying.
No, I'm paying for hers. All right, I hit salary, right?
As he said, I'm Bernice Moser, and I'm doing the estrogenic effects in native inurans and local watersheds. I'm going to dim the lights now. Amphibian populations across the world are thought to be in decline. This has been contributed to several factors, including climate change, pollution, increased ultraviolet B radiation, and disease. Often these factors are talked about in terms of mortality. However, there are many chemicals in the environment that only impact sublethal effects such as growth and development, behavior, and reproductive capability. One category of chemicals that affect reproductive capability is called reproductive endocrine disruptors. These compounds mimic or inhibit naturally occurring hormones, such as estrogens and androgens. And with estrogenic chemicals in particular, they encompass a wide variety of chemical classes, and they have a weak structure-activity relationship, which makes them very difficult to detect in the environment. And you add to that depending on the compound, it can have a different affinity for binding the estrogen receptor in an organism. This can vary as much as 2 to 3 orders of magnitude. Microflora in the environment also alter these estrogenic compounds either into more active or less active forms, which complicates things even further. The compound that you see here is 17-beta-estradiol. This is the compound that regulates reproduction in many classes of vertebrates and in all classes of invertebrates. These other compounds are known to weak estrogenic disorders. There are a couple of organofluoral pesticides, and they all are known to act about 2 to 3 orders less magnitude than the estradiol does. And you can also note with these structures the rarity of chemical structures that you can have with estrogenic compounds. There are many sources of estrogenic disruptors, including agricultural, industrial, and wastewater effluents, pesticides, and pharmaceuticals such as birth control pills. In agriculture, the hormones that are released by cattle and poultry naturally are augmented by excess steroids such as trembolone acetate, nonogasterol acetate, and nonogasterol acetate. Cattle urine itself has been shown to have E2 concentrations of approximately 13 nanograms per gram, and poultry dry waste has been shown to have E2 concentrations of approximately 44 nanograms per gram. These compounds enter the aquatic environment from runoff and reapplication of waste as fertilizer, because there are many countries in which agriculture or cattle waste are reapplied to the land as fertilizers. Industrial wastewater effluents also have several compounds that are known to be estrogenic, including surfactants such as 4,9-phenyl. 4,9-phenyl has been shown to have estrogenic effects in male manila clams and in Atlantic salmon. Plasticizers such as bisphenol A have been shown to be estrogenic in the South African cod frog, Xenopus laevis, and in male killifish. Polychlorinated biphenyls have actually been shown to be both estrogenic and anti-estrogenic. These compounds competitively bind the estrogen receptor, where they can either act as an estrogen agonist or an estrogen antagonist, meaning that either they can bind the receptor and induce the response that estrogen has, or that they can bind the receptor and prevent estrogen from binding and thereby inhibit the genes that estrogen activates. PCB conjugators 101 and PCB 70 have been shown to actually alter the 50-50 sex ratio in the male northern leopard frog or amphibians. Pesticides such as atrazine, which is a very commonly used pesticide, has been shown to suppress testosterone and 11-ketotestosterone and has resulted in elevated levels of 17-beta-estradiol in the adult goldfish. Organochlorines such as methoxychlor have been shown to have estrogenic effects in zebrafish, and pyrethroids such as cypermethrin have been shown to have estrogenic effects in softtip. Pharmaceuticals that are found in both human and veterinary medicines enter the aquatic environment in sewage and wastewater effluents. These include the natural estrogens, estrone or E1, 17-beta-estradiol or E2, estriol E3, and the synthetic estrogen 17-alpha-ethyl estradiol EE2 and mestranol ME2. Both EE2 has been shown to have a higher binding affinity for the estrogen receptor than E2, and ME2 has a lower. Both of these compounds have been shown to be more resistant to biodegradation in sewage fluids by the microflora. And in actually an aerobically treated sewage has been shown to have little to no estrogenic and androgenic activity, while anaerobically treated sewage has been shown to have both estrogen and androgen gene transcriptase activities. Because of the difficulty to detect them, these compounds are often identified through their biological activity, and one method of doing this is to measure a biomarker. These allow for the detection of specific physiological or cellular effects in response to specific xenobiotics. Biomarkers can reduce the cost of chemical analysis, and it allows for the detection of complex chemical interactions in the environment. One such biomarker that's used with reproductive endocrine disorders is vitellogenin. It has been used in both vertebrates and invertebrates. It is the precursor to the yolk protein in oviparous females. It is not normally produced in males and juveniles, as circulating levels of estrogen aren't high enough to stimulate its production, but it can be induced by estrogenic compounds. Vitellogenin is often detected through methods such as ELISA and Western blot and alkyl labile phosphates. ELISA and Western blot are generally considered to be more specific and more sensitive. However, because of the necessity of developing antibodies, this can be more costly in time and expense. And you're also limited to specific species that the antibody will cross-react with. Assays such as the alkyl labile phosphate assay tend to be less sensitive than Western blot or ELISA, and more often than not, they require a much larger sample volume. Recently, an assay has been developed that can be applicable across species. It does not require the development of an antibody, and it does this through the use of a commercially available dye called protein phosphoprotein gel stain, which detects phosphorylated proteins as serine, tyrosine, and threonine residues. It has been shown to be as sensitive as Western blot analysis, and it allows for low sample volumes. It has been successfully used in the mummy chub, in fathead minnows, and in mosquito fish. It has not up until this time been applied to amphibians. Xenopus laevis is a well-studied amphibian model, and its physiological and reproductive and endocrine systems have been thoroughly documented. It can be induced to breed year-round and produces large quantities of eggs. It is entirely aquatic, making it an ideal in vitro